Hello, welcome back to day 2 of viral pathogenesis. So I hope y'all are having a great day. And today what we're going to do is we're going to pick up where we left off. So last time we talked about this idea of pathogenesis versus virulence. We talked about what affects viral pathogenesis and how we study human diseases. And we finished up part 1, which is patterns of infection. So today we're going to start with part 2, and that is viral virulence. So in day 1, we define virulence as the degree or intensity of pathogenicity. So let's talk about what a virulence factor is before I send you off to do this brainstorm practice problem. So a virulence factor is any trait that a microbe has, and these traits are going to help them as a pathogen. And so these virulence factors are going to affect virulence. And before I send you off to have you pause the video and to brainstorm and work on this practice problem, one thing that I want to say is much of what we know in terms of virulence factors is from studies of attenuated viruses. So attenuated means reduced virulence. So you have probably heard of the terminology of attenuated when people use it in terms of attenuated vaccines. And when we talk about vaccines, we'll actually come back to this idea of attenuated viruses being used as vaccines. But for now, what we need to know is that attenuated is reduced virulence, and using attenuated viruses, that's how we learned about many of these virulence factors. So go ahead and, based on what you know so far from this topic, pause the video and see what you can come up with. All right, so hopefully you've gone ahead and jotted some things down, and I'm going to share some things with you, and if there are things that I have listed that you don't have listed that I do, go ahead and write them in the comment section below. So we can have a lot of things that can help contribute to virulence as a whole for viruses. And if you've ever played the game Plague Inc., they have really great examples of virulence factors in there as well. And so that's going to be things like transmission. So is the virus able to transmit? Oftentimes we say things like fomite, which is an inanimate object. Is it able to be airborne? So again, during day 1, we kind of talked about this idea that people are asking, is the coronavirus airborne because of how easily it's being spread? So transmission is definitely something that is going to be a virulence factor that could help a virus as a pathogen. So other things are going to be receptors. So when we first started talking about the viral reproductive life cycle, we started with how does a virus recognize its host? And so having receptors, especially receptors that have fluidity or plasticity, may allow for things like escape mutants, or may allow for viruses to have alternative receptors, so that they're able to use maybe more than one way to gain entry. So all of these things are going to help a virus be a better pathogen. So first, other things are going to be how hardy is the virus? So how hardy is the virus is referring to things like what can it withstand? Can it withstand temperature changes, pH changes, pressure changes? So we've spent some time in this class talking about how naked viruses are definitely much more stable in the environment than enveloped viruses. So one of the things we have going for us in this pandemic is that the coronavirus is enveloped, and so it can't stay in the environment for as long as many other viruses can. And of course, there are many other examples as well. So tropism, so this idea of what is the host range like? What is the cell range like for the infectiveness? How invasive is the virus? How fast can it replicate? What are some of the strategies that it uses to replicate? And for a lot of the examples that I've come up with and the ones that you can probably come up with, they fall underneath a couple of different categories. And the categories for virulence factors are going to be things that help a virus colonize the host, So we talked about how all pathogens first have to get in. So colonizing the host, how do they enter? So things like immunoevasion. So another thing that all pathogens have to do is they have to fly under the radar. They have to somehow avoid the immune system. Sometimes some viruses also participate in immunosuppression. So we'll talk about that as well. Of course, somehow we have to have obtaining nutrients from the host. So we've kind of started to hint at some of those things. So when we talked about how viruses modify the different metabolic pathways that are normally found in cells, that could absolutely be an example of a virulence factor as well. So I specifically want to talk about two really cool examples. And the two examples that I want to talk about are called Virokines and viroceptors. So let's start with Virokines and then we'll work our way to viroceptors. So viruses can be pretty sneaky, and what a virokine is, this is a secreted protein, and this is a secreted viral protein. And what this does is this mimics cytokines and growth factors, but it does not transduce signals. So what do I mean by that? Let's first look at how a cytokine works. So a cytokine is a small protein, and it's a signaling protein that is released. So say we have a cell and maybe the cell is damaged somehow, it's infected. So let's go ahead and draw a little virus in here. This cell is going to produce a cytokine. That cytokine is going to travel. Eventually, it will reach another cell. Cytokines typically signal our immune system. That cytokine is going to be bound by a receptor, and this is going to signal for a response. Usually, in the case of what we're talking about, this response is going to be to turn on the immune system. And so we're going to get a series of things like inflammation, fever, all of the other components of the innate immune system that are turned on. So what a virus does is it actually encodes for proteins that instead of the cytokine binding the receptor, so here's that receptor, let's add a signal pathway here. Instead of the cytokine binding the receptor, a signaling pathway happening and a response happening, what a virokine does is the virokine will go ahead and it will bind that receptor, but we get no signaling pathway. And so it mimics a cytokine in how it works in terms of binding to the receptor, but not in terms of the signal transduction pathway. And so it's going to stop that as well. So another really great example of a virulence factor that viruses have are viroceptors, and viroceptors are going to mimic cytokine receptors, and what they do is they divert signaling molecules from finding targets. So if we have the same cell that is infected by a virus, and one of the proteins that it has made is a viroceptor, when this cell recognizes that something is wrong and a cytokine is sent out, and that cytokine is meant to be a signal to the immune system that something is wrong, instead there's a viroceptor really close, and so if we have these viroceptors, these cytokines can then interact with that, and essentially it's just a really good way to stop that signaling pathway that we drew here on the left from occurring. And so these are examples of ways of immunoevasion, immunosuppression, and they're some of my favorite examples of virulence factors that viruses have. And I think it's just really cool and really amazing. So kind of moving forward, in terms of, well, what makes a pathogen virulent? Why is one more virulent than another? So what I want you all to do is to go ahead and, again, brainstorm some things that you think is going to help make a pathogen more or less virulent. So I'm going to share some things with you, and again, in the comments below, let me know if I left something out. So in terms of virulence, something that could be really helpful is a large host range. So if you are only able to infect one type of host, and that host is not very common, you're probably not going to get as many progeny. And so if you have a larger host range, higher tropism, and that could be cells in the body, or that could be looking at organisms itself, that's going to be beneficial. Perhaps high progeny, so thinking about having acute infections, and when we think about those R strategists that we have, their strategy is this idea of making a lot of progeny, not investing a lot of energy, hoping something hits, and so they tend to have things like high mutation rates. So maybe a high mutation rate is a good thing as well. So they're able to, because of these high mutation rates, maybe you're lucky enough to get a mutation in just the right spot that helps you have a stronger binding affinity towards your host receptor, allows you to better evade the immune system, so on and so forth. So things that are really hardy also. Maybe perhaps if we think about this idea of not killing the host. Again, the goal of viruses is not to cause disease. The goal is to reproduce. But if you can be pretty virulent and not kill the host, maybe that gives you an edge, because this leads to this idea of immunoevasion. And then first we can also add this idea of easy transmission. So the easier it is to transmit to your host, the more likely it is that you're going to be able to attach and enter and reproduce. So again, let me know in the comments below if there are other things that you came up with. So of course there are different metrics of looking at these things, and two of the metrics that people look at to help to answer this question of which pathogens are the most virulent is so we have two things. We have the lethal dose 50, and this is also referred to as the LD50, and we have the infectious dose 50 or the ID50. So we're going to define these, and then we're going to apply it to this question. So the lethal dose 50 is the number of infectious particles that will kill 50% of the host. So the number of infectious particles that will kill 50% of hosts is known as the lethal dose 50. And although we're talking about these two terms from the perspective of virology, these can also be expanded to include other pathogens as well. So for bacteria, we would say the number of bacterial cells that would kill 50% of hosts. So again, these are terms that are used pretty broadly across microbiology and virology. So in terms of the infectious dose is the number of infectious particles that will establish an infection in 50% of challenged hosts. Okay, so knowing what these two things mean now, I want you all to think about what would make the most virulent pathogen. So if we have a virus, the most virulent pathogen is going to be the one with the highest or lowest LD50 and the highest or the lowest ID50. So again, hopefully you've taken the time and you've worked through this. And for this practice problem, the most virulent virus is going to have the lowest LD50. So we're thinking about virulence, of course, from the human perspective. So this is seen as something bad, lots of people getting sick, lots of people dying, not a lot of viral particle is needed to get you sick. And so the lowest LD50 and the lowest ID50 is going to be the most virulent virus. And if we were talking about this in microbiology, the same would be true for bacteria, for fungi, for parasites, etc. Again, this is something that is very broadly accepted across different fields of microbiology. So if you have questions on that, please let me know in the comments below, and I will do my best to walk you through them. So now that we've kind of talked about this idea of virulence, we've introduced virulence factors. So we've talked about how attenuated viruses are how we've learned a lot about these virulence factors. Using that information, I want you all to brainstorm and think about what are some experiments that we could do to measure viral virulence. Okay, and because this is a question that has a lot of right answers to it, what I'm going to do is I'm going to tell you the categories that virulence genes can fall into and give you some general examples of what those are, so kind of expand upon what I've already said. And then I'm also going to tell you kind of some general ways to measure viral virulence. And then as part of the discussion for this, in the comments below, I would like each of you to either come up with an experiment that you could measure viral virulence with, jumping off of what I have said, or if someone else comes up with an experiment in the comments that you think is interesting, talk about in more detail how that experiment could be implemented and what could be done. So when we have virulence genes, they're going to fall into a couple different categories. They're going to fall into categories that affect the ability of the virus to spread. They're going to fall into a category of potentially modifying the host's defenses. We can also have things like facilitating viral spread. And this could be viral spread in and among different hosts, or these can be things that are directly toxic. And some examples, of course, these are going to be things like our virokines, our viroceptors. So those are specific examples that we just talked about. But these examples can also include genes that encode for or perhaps force the cell to enter the cell cycle. And we'll talk about some of those in the next topic. So that will be covered in Oncogenesis and how viruses can potentially accidentally lead to cancer. And if you think back to the infected cell, this could be genes that also change metabolism. So think back to the hepatitis example that we talked about. So what I would like you to do is pick a gene that you think that a virus could encode for, or a protein product potentially that we've either talked about in this class or in the research that you've been doing for your virus project, and use that to draft your experiment. So let me give you a couple of hints and a couple of things that you could look at. So again, I'm not going to walk you through all of the experiments because we would be here for a really long time, but I want to give you a starting point for this. And one of them is viral titer. So we've done this already in lab. So think about what we did in lab before break and how a virus titer might help us determine viral virulence. We can also look at time to death. So if the lethal dose 50 is known, so maybe somehow we could tie in lethal dose 50 and tie in time to death. So let me jot down LD50, ID50. Both of these would somehow help us. And again, there's lots of different ways that you could look at these numbers. So this idea of time to symptoms. So looking at the incubation period. So you could also do things like measuring pathological factors. So in HIV, we see a decrease in CD4+. We see lesions in some viruses. In coronavirus, we see the lesions in the lungs, and so that would be considered a pathological factor. So we can potentially somehow measure that. And as you're thinking about ways to determine virulence factors or these genes and how we would measure virulence factors, I want you all to be thinking about how you can look at this. So you can look at this at the cellular level. You can also look at this at the organismal level. You can also look at this at the host level. So when I say organismal level, maybe you're looking at the different model animals that we can use to study this virus. And when you're thinking about this second question, which is how do you determine virulence factors and genes, think back to a practice problem that we did near the beginning of the semester where I asked you all to determine what a potential protein that a virus uses as a receptor protein could be. So think about some of those knockout assays that we talked about. And we can also kind of think about sequencing in here. So I've given you a lot of hints for these two different experiments, and I'm really looking forward to the discussion that we'll have in the comments below. So let's move on to part 3. So part 3 is pathogenesis. So we said that pathogenesis is the mechanism of causing disease. And disease, of course, is a negative change in a person's health. It could also be referring to damage to the host. And so the question is this idea of, well, how is the pathogenesis of a virus related to its ability to infect a host? And this really comes back to this idea of using these different virulence factors or these virulence traits to cause disease. So if we kind of expand upon that, the question becomes, well, what are the different kinds of things that viruses can do to cause disease? And remember, disease is just a very broad definition of a negative change in a person's health, any damage to the host. So if we think back to killing cells, well, that's damage, right? Doesn't that lead to destruction? Again, we probably don't realize when only a couple of cells are destroyed, but that is still considered disease. And a really great example of proteins that do this is viroporins. And specifically one that we've talked about in this class is adenovirus death receptor. And Ben actually said that this is his favorite protein. So and I think I said adenovirus death receptor. I meant adenovirus death protein. So adenovirus death protein is Ben's favorite protein. So this is a really great example of something that viruses do. And again, this is part of their normal reproductive life cycle, but this can cause disease. And we can pretty much think of any change that is molecular or functional or structural to a cell. And then, of course, eventually the tissue is considered causing disease. So examples of that are going to be rearranging our organelles. So we saw in the infected cell that many viruses completely remake what the Golgi and the ER looks like. They make those viral factories. Those are part of how the virus reproduces, but that is causing damage to the host. This can also be the nucleus. So we see the nucleus changes. This can be changing metabolism. So, of course, in the case of the hepatitis viruses, we talked about how prolonged changing metabolism because of these viruses can lead to type 2 diabetes and can lead to fatty acid disease. So all of these things are definitely molecular, functional, or structural changes that happen to a cell, and they are all part of causing disease. And of course, we can add our idea of immunosuppression up here. So using those virokines and viroceptors. Another thing that we've hinted at, but again, we will have an entire topic on this, is oncogenesis. So oncogenesis is causing cancer. So I think I mentioned in day 1 that 20% of human cancers are associated with viral infections. So we'll talk about that. And of course, we have a lot of things that would fall under this idea of immunopathology. And immunopathology really just deals with this idea of critical signs and symptoms of viral disease. And that's going to be mostly things that our immune system is responsible for. So fever, aches, pains, inflammation, all of these things are part of the normal immune response to antigens. But because our antigen is a virus, viruses, of course, are the causative agent behind this. So again, this is just kind of meant to help us tie together this holistic view that we're taking of pathogenesis and virulence. So what we're going to move on to now is this idea of susceptibility. So we can have a virus transmit, we can have a virus enter and replicate, but what really matters is this idea of susceptibility. And so what I want you all to do is to brainstorm and to think about what's going to affect whether or not a host is susceptible. And I'm going to share some of the things that I've come up with, and then let me know in the comments below if you have other things to add to this. So one thing that we can think about is the receptors. We know that all viruses have a specific receptor, sometimes more than one, that they require for gaining entry into the host. So whether or not a host has a specific receptor is going to help determine whether the host is susceptible. And of course, the immune system. So is a person immunocompromised or not? This could potentially affect whether or not a host is susceptible, depending on the virus. Like thinking back to what we talked about in day 1 and coronavirus, of course we can have biological sex. Sometimes we see differences there. We can throw genetics up here. Genetics, of course, might control which receptors are there. It might control the number of potential receptors. We could have other diseases. So in the case of the current coronavirus outbreak, we see that, people of all ages are getting sick, but overall we still see more complications with people that have other underlying diseases. So heart problems, diabetes, things of that nature. And a lot of that goes back to the receptor usage of this virus. And we're also seeing, of course, people who seem to be getting sick and they don't know that they're getting sick. So they are still susceptible, but for whatever reason, they just are not having the same symptoms as everyone else. And while we still have a lot of experimentation we have to do and a lot to still learn about this, this is common with many viruses. So again, there are a lot of things that are going to affect which hosts are susceptible. So the first example that we are going to talk about is HIV. And many of you have probably heard of the Berlin patient. And the Berlin patient actually refers to 2 patients. The original Berlin patient is anonymous, and the second Berlin patient actually came out and said who he was. And so we're going to focus mostly on the second Berlin patient, but keep in mind that this also applies to the first Berlin patient as well. And so our second Berlin patient is Timothy Ray Brown. And so he was diagnosed with HIV in 1995. And in 2006, he was diagnosed with leukemia. So he was diagnosed with cancer. And during this time, his doctor had a hypothesis, and he was, I guess, lucky enough that Timothy Ray Brown kind of went for it. And so one of the treatments for leukemia is a stem cell transplant, so with hematopoietic stem cells. And so his doctor had a hypothesis that he wanted to test. And when they were looking for donors for the stem cell transplant, they were looking for a particular donor. And the particular donor that they were looking for the stem cell transplant was someone that had something known as a CCR5 delta 32 mutation. And the delta means deletion. So you're probably, hopefully, scratching your head like CCR. That sounds familiar, and it should. So when we talked about HIV, we talked about the two receptors that HIV uses. So we said that GP120 requires CD4, and so when it interacts with CD4, it then needs a co-receptor, a CCR. Usually it's CCR5. And upon that co-receptor recognition, that's when we actually see entry that can occur. So for this Berlin patient, for both the first and the second one, the doctors specifically chose patients that had a deletion in CCR5. So if this is what a normal typical CCR5 looks like, we've got our receptor here. So here's where the binding portion of the protein is. CCR5 delta 32 is missing 32 amino acids, and it's missing part of this portion on top here that would be recognized by HIV. And so from this, his doctor hypothesized that maybe this would somehow help, because if he's getting a stem cell transplant, stem cells are what give rise to progenitor cells. And so this idea then is maybe then his cells wouldn't have CCR5 or be missing that little tip, right? And because they're missing that tip of CCR5, HIV no longer has the co-receptor that it needs. And so it turns out that after this stem cell transplant, they no longer detect HIV in his blood. And he's actually no longer on antivirals. And so oftentimes people consider him cured. So of course, cured is kind of a loosey-goosey way of playing around with it, because you and I know that HIV integrates into the genome, and so he still has HIV genome integrated into his genome. And so, you could argue that that means he is not cured. However, he is no longer producing progeny virions, and they can't detect the HIV in his blood. And so it turns out that about 16% of Europeans have this mutation. So it's not crazy common, but it's common enough where we have determined the percentage of the population that has this mutation. And if you're wondering why, we think maybe it has to do with smallpox. And we think that perhaps this receptor was also used by smallpox. And so Europeans that survived the smallpox pandemic were ones that happened to be missing part of the CCR5. And last year, so in 2019, early 2019, we have who we refer to as the London patient. And the London patient actually also revealed who he was. And the article that is on this page here, this article is about him and essentially a very similar story to Timothy Ray Brown. And so Adam got diagnosed with HIV, and then he later developed Hodgkin's lymphoma. And when he developed Hodgkin's lymphoma, he also had a stem cell transplant. And when he had a stem cell transplant, his doctor wanted to duplicate the procedure that led to the long-term remission 12 years ago, which was Timothy Ray Brown. And so they also were able to find a stem cell donor that also had a CCR5 delta 32 mutation. And it appears that this patient is also now no longer on antivirals, and they can no longer detect HIV in his blood as well. So, of course, this is very, very expensive procedure, and we have a lot of work to do kind of moving forward in terms of, is this a viable treatment for patients with HIV or not? But nevertheless, both very cool stories of I'm going to say curing, in quotations here, patients. So I wanted to share that, and this idea of susceptibility directly correlates to these examples of the Berlin patients and the London patient, because both of them now have a receptor that is shortened, essentially. And of course, we still have a lot of work to do with this, and you have probably seen in the news that there was a scientist in China who actually used CRISPR-Cas, and this is a gene editing system. And what he did was, there was a patient that was pregnant with twins, and using CRISPR-Cas, he went ahead and essentially trying to, in a way, kind of duplicate this experiment, but at the genetic level in these twins, removed part of the CCR5, and I believe that the twins were born, but we don't really know anything else about it because much of it has been kind of hush-hush. So, again, more of an ethical debate, but definitely something that ties back to this idea of susceptibility. And much of this was done because I cannot remember if it was both the mother and the father that had HIV, or if it was one of the parents, but nevertheless, it goes back to this idea of susceptibility. So if we can remove the receptor, if we can somehow stop being susceptible to a virus, then we don't have to worry about it causing disease. Now, the second example that I want to talk about is our microbiome. And this is, I think, a really, really cool example. And it's amazing to me, and definitely something that was very new to me. Before I started teaching this class, I never really had thought about how our microbiome can affect the ability of viruses to infect us. So we, of course, have a microbiome, we have a virome, but I never really thought about how they could play and kind of feed into one another, which is, of course, silly because they normally exist together and whatnot. So let's first kind of think about the microbiome. So the microbiome just deals with who is there and in what quantity. So we have actually looked to see what microbes are normally found on our face, in our armpits, in our groin, on the inside of our intestines, so on and so forth. And typically we think of our microbiome as our microbiome helping humans, right? So definitely a protective mechanism in some cases. And so without knowing much about the microbiome, I want you all, before I tell you this story, to tell me your thoughts and ideas of how you think our microbiome might affect the ability of viruses to infect us. So go ahead and do that, and then I'm going to put up a blank page, and then we're going to talk more about this. So I ran across this article last year and it really sparked my interest. So this article is titled Intestinal Microbiota Promote Enteric Virus Replication and Systemic Pathogenesis. So I just told you that the idea is that maybe our microbiome helps protect us, right? It helps humans. In the case of digestion, absolutely. The microbes in our digestive tract definitely help us digest our food. We have E. coli that live there that make vitamin K for us because of course we can't make our own. But this was a really cool example of, wait, that's maybe not always true. So what this paper did was these scientists looked at poliovirus and they looked at reoviruses. And they completed this work with mice. So again, mice are model organisms, and we talked about how mice lie and monkeys exaggerate, but nevertheless, it's still a really cool paper and a really cool experiment. And they had two groups of mice. So they had mice that were not treated, and then they had mice treated with antibiotics. So the mice that were not treated were first the mice that had their normal microbiome. The mice treated with antibiotics, when we take antibiotics, it not only kills the bad bacteria, kills the good bacteria too. So it got rid of their microbiome. And then after those mice that were treated with antibiotics had their microbiome cleared out, they gave polio, or they infected with polio. So when they infected with polio, they did this to both of the groups of mice. All right, so what did they see after they infected it with polio? So in the first group of mice, they saw that more were sick. They saw lots of viral replication, and they saw a higher death rate. So about 2 times higher than that of the mice that were treated with antibiotics. So what they saw in the mice that were treated in that treatment group, they saw that these mice were less susceptible and there was less viral replication. So those were the first 2 groups. So then what they did was, okay, well, what happens if you then take these mice that were less susceptible, had less viral replication because they were treated with antibiotics, and you reintroduce fecal bacteria? So what they did then was they took mice treated with the antibiotics, and then they reintroduced fecal bacteria. And we actually do this for patients that have very severe cases of C. diff. We will reintroduce fecal bacteria through fecal transplants. This process is definitely something that is argued about, both clinically speaking and then also in the literature as well. But in the very extreme cases, this has been done as a means of trying to reintroduce the normal, healthy microbiome that is there. And what they found was when they reintroduced those fecal bacteria, they saw enhanced polio disease. What their conclusions were, and again, I should reiterate that the same set of experiments was completed with both poliovirus and reoviruses, and they saw very similar results in both. So what their conclusions were was that something, right, so those bacteria in our gut are helping viruses, which is kind of amazing to think about because the viruses that they're working with are, of course, eukaryotic viruses, which do not infect bacterial viruses. And so their other conclusion was that enteric viruses have evolved to use an intestinal microbe as a trigger for replication and transmission. So these viruses exploit enteric microbes for replication and transmission. And if this doesn't make you want to be a microbiologist and study virology, then I don't know what does. But this is a completely and utterly amazing example of something that is normal found in humans. So normally found in us. We all have a microbiome. Our microbiome, it helps us. Our immune system, it has learned to decrease its attack against those microbes because they are beneficial. And here we have these pirates of cells that are going to hijack our cells that have somehow evolved to use what is supposed to be helping us as a signal for transmission and replication. So to me, this is just absolutely and utterly amazing. So this actually wraps up our discussion of pathogenesis. And again, I hope that I have convinced you that this idea of virulence and pathogenesis and susceptibility and patterns of infection really kind of comes together for a holistic view of infection of viruses. And although we like to separate them, and they are really important to look at how they affect one another as well. So in the comments below, leave any questions, comments, any other things that you want to add to our discussions, and I will catch you in the next video. And in the next topic, we are going to talk all about viral transformation and oncogenesis. So have a great day, everyone, and I will see you soon. Bye, everyone.